So this is Dr. Marin Sawa from Newcastle University, a NUAC fellow at Newcastle University within the Hub for Biotechnology and the Built Environment. And her talk today is Biodesign, Designing with Microbes to Devising Applications. Over to you, Marin.
Hi, thank you very much. So I'm a new addition to HBBE, and can you see my screen? Can you
see? Yes, I can. Yes, we can.
Okay, yes. And yes, I'm really excited to really explore interdisciplinary research that I've been doing in the field of biodesign. And biodesign, as we all know, it is about integration of biological system into design. And the essence of my research really in this field focuses on different cell cultivation methods, ranging from the left, encapsulation of cells in alginate, to the designing of photobioreactors based on liquid culture, to biofilm bioreactors based on solid cultivation of cells. And the last 4 or 5 years been spent on developing this solid culture system for bioelectricity generation. And in collaboration with a team of scientists at Imperial and Cambridge, I sort of innovated a new system for this biophotovoltaic technology, which was published in Nature Communications. And I've been engaging at a higher technology readiness level, trying to find feasible applications for this very emerging technology. And I exhibiting, for instance, at IDTech X, an emerging technology fair host in Silicon Valley, and sort of introducing this biological system to a new field such as printed electronics. So I've been working with photosynthetic microorganisms as my biology to really design with. And as Shafir already explained, photosynthesis really can be described in different ways, mainly on the water splitting by right reactions. But I would say that algae and cyanobacteria utilize light energy to convert water and carbon dioxide into carbohydrates to feed themselves. And in the process, especially in oxygenic photosynthesis, it liberates oxygen into the atmosphere. And as you must have seen algae everywhere in our environment. They do grow given all these prerequisites. And they grow in both aquatic environment and terrestrial environments. And these are actually translated into how they are cultivated in research and industry, more predominantly liquid culture for large scale up. But emerging works have been done on solid culture since 2000. Now, why am I interested in algae? Well, algae are, besides a crucial role in using sunlight to fix atmospheric CO2 into biomass and oxygenating our atmosphere, algae, both marine and freshwater, involved in global cycling of nitrogen. And these four sort of crucial ecological contributions are translated into applications, as you can see on the bottom, for human uses. However, except for nutraceutical and some chemical productions, these applications have not reached commercialization. And mainly because it's very expensive to grow algae, and the costs involved do not justify agrobiotechnology applications. However, the algae have been part of our human history and in a built environment. And the development of photobioreactors in particular helped architects and designers not only envision, but also materialize agrobiotechnology integration into the built environment. And this was really nicely described by Barbara this afternoon. And my research really is looking at the integration of agrobiotechnology into domestic spaces and social practices. So coming back to the how I would integrate my cells into the built environment, more precisely into electronics, I'm looking at how cell seeding is done in biotechnology. So there are two modes, passive and active. And the passive mode utilize natural cell attachment by submerging them in liquid culture where cells are suspended. And the cell attachment to the substrate is then incorporated into a device design such as biosensor. In the active mode on the bottom, it uses mechanical forces to deposit cells directly onto electrode surfaces. And this really provides a greater degree of control over where cells grow and where cells shouldn't grow. And much has been explored in the tissue engineering field with mammalian cells. Actually, I apply this method to printing photosynthetic microorganisms during my PhD back starting from 2012 onwards. And I was the first person apparently to do so, surprisingly. And the characterization of this method has been successfully done, and printed cells show very good cell viability through on the bottom, and also the to be actually precise, 98% of printed cells are viable. And the photosynthetic competency of the cells is quite separate from cell viability, and this can be analyzed through chlorophyll fluorescence imaging using PAM, pulse amplitude modulate barometer. And the FV of FM, which is the parameter for maximum quantum efficiency of photosystem 2, was found to be 0.4 of my printed cyanobacteria, Synechococcus synecocystis, on paper substrate. And this 0.4, this value really is in good agreement with that of liquid culture. So you can see that the importance of keeping cells alive on substrate and being verified through application of science in my research. And my research really explored this inkjet method to fabricate an entire VPB system. And so layering of the biology onto substrate is one thing, but layering conductive conductor onto the substrate is another. And the same inkjet method, which is actually a thermal inkjet printing using heat to jet the materials from the nozzles, was applied to deposit carbon nanotube conductor, as well as the carbon-based catalyst for and this really was the novelty of my inkjet method, allowing biological and non-living layers to be fabricated in the single process, but in different steps. The important thing is that the cells then grow over this carbon nanotube interface. And this is the mechanism of the printed biophotovoltaic system, whereby photosynthesis generate electrons by water splitting and generate liberating electrons and protons, allowing them to migrate to the cathode for oxygen reduction reaction. Oops. And this is the design of the BPV system by inkjet printing, which allows greater freedom of design over where the cells are deposited, and the zigzag patterns were created through the printing capability. And also, it sort of allows greater versatility in terms of which materials these cells can be deposited, and also offered some solution to a scalability issue in BPV research. Now, the capability of this printed BPV system, it can be found here. So the power output of the fully printed system, which I characterized and developed with Andrea in the, as you can see in the lower left-hand side, it shows the production generation of electrons over 4 or 5 days in the range of microwatt. In the light and in the dark. On the right-hand side, the improved hybrid system was developed in collaboration with Paolo Bombelli at Cambridge. And this really incorporates more effective catalyst, platinized carbon paper, but it includes heavy metal, the platinum. So that was not really my intention. However, for the optimum power generation, this proved to be 30-fold more effective in terms of power output. So the microwatt range to the milliwatt range was achieved. And with this hybrid system, even though not fully printed, we demonstrated the powering of something useful like a digital clock here over the period of 3 hours. However, you can see there's an intermittent rest from the cells every half an hour. And this is one of the limitations of this kind of biological system for electricity generation. My focus actually has been how really to keep cells intact and alive and healthy and form really mature biofilm over a conductive surface. So on the left, you can see my printed biofilm forming a really, really thick layers. And this is compared to a more conventional cell deposition through the passive mode. On the right, it's clear that the cell printed system is more effective, achieving really high cell density. And on the bottom, you can see the real sort of interface between the paper fiber and then the carbon nanotube, which is creating this kind of mesh texture, and how the cells, in this case, Synechococcus, with the sort of tubular forms, are actually attaching themselves to the electrode surface. And throughout this HBB summit, I've heard different approaches as to how we might deposit cells. And I would say that there is also a biological approach to how we might enhance biocolonization on different materials. So I explored different species. One is Lectolimbia ohadi, desert cyanobacteria, which forms really robust biofilm excreting EPS, which I hope you can see here in the middle of this slide, which and the EPS production is very distinct, especially compared to Synechococcus biofilm on the right. And electron generation and tolerance to desiccation can be species dependent. Therefore, depending on which species you use, you can actually achieve different level of bioelectricity generation and different levels of tolerance to desiccation. And tolerance to desiccation, in my case, is really important because this printed system is actually an open system rather than closed system. And the desiccation of cells is one of the critical issues I would need to solve. And I'm hoping to do this from a biological perspective, as well as sort of material science perspective, in addition to forming interrelations with our environment. And by exploring this printed system, there was a new application which was which mobilized really enthusiasm in the public. And I'd like to just play this video quickly, which was created by World Economic Forum, and really sort of connecting how this could be applied to our domestic spaces. So one of the virtue of this system is it's biodegradable. Well, almost. The biodegradability of carbon nanotubes is still debatable, but it shows some promise to tackle our global e-waste crisis. And my research really is looking at a so-called ecosophical approach, connecting the environment, society, and mind, and at the intersection of these three domains lies how we engage with social practices. And my research tries to really position this emerging technology to engage with these three domains. And I'm looking particularly for this New York Fellowship to integrate paper-based architectural components in Japanese architecture, such as shoji screens, to be the testbed of the integration. I'd like to thank my collaborators in the past, particularly Peter Nixon, Klaus Helgert, Andrea Fantuzzi, Paolo Bomberi, and Cristal, who are the co-authors of the paper. And I'd like to thank my previous funders, including Central St. Martin's for my PhD scholarship, and Imperial College for EPSSC and the Texelerate Fellowship, EPSSC Acceleration Account Grant, and also Texelerate Fellowship. Yeah, so any questions, please ask me.
Thank you, Marin. Unfortunately, I don't think we're going to have enough time for questions, but again, if people could put questions into the chat, then if you could have a look at those, Marin, that would be really good. Thank you for that. Thank you.